Hello, today we are going to explore Chapter 2, the chemical level of organization within the body. So let's look, how is matter organized? First, we're going to look at the chemicals, or the chemistry, which is the science of the structure and interactions of matter. So today, most of the lesson is about chemistry. There's a little bit of physics, but it's mostly chemistry. And when we're talking about chemistry, we're always talking about mass and weight. The mass is the amount of matter in any object. That does not change. Doesn't matter how big or small, mass is always the same, while weight is the force of gravity acting on that matter. Weight changes. Just like, say if you were to step on the scale every single day, your weight changes little by little because that's the amount of force that gravity is pushing down on you. Or the structure of the different molecules. We have the mass, how much it weighs, and we also have the weight. Weight is the force of gravity. So, like the weight you see on the scale, that's the force of gravity pushing down on you to create whatever weight you see. Weight can change, as you know, if you step on the scale often enough. Every day it's a little bit different, but the mass of an object, that does not change. Mass is always consistent. Matter is also thought of in 3 different states: a gas, a liquid, or a solid. Think of water. Water in a gas is like fog. Or steam when you're boiling water. A liquid, water is a liquid. A solid, ice. Some other examples: your bones are solid. Teeth are solids. Well, the plasma in your blood, that is a liquid. And again, oxygen and carbon dioxide are gases. So in our bodies alone, we have 26 different chemicals. 96% of the body's mass is actually made up of oxygen, carbon, hydrogen, and nitrogen. While 3.6% of your body's mass is made up of calcium, phosphorus, potassium, sulfur, sodium, chlorine, magnesium, and iron. And there's always trace elements found in the body, other types of chemicals. Those only make up about 0.4% of your weight. There's 14 total, but the main one really is iodine. I have a chart here to show, again, here's our major elements, and we look at each major element like oxygen. It's part of water. It's part of many carbon-containing molecules. It's used to help generate ATP for energy. Carbon forms chains for molecules. Carbohydrates, we'll look into what those look like. Lipids, proteins, nucleic acid, which is your DNA and your RNA. Hydrogen, that's used to make water. There's also hydrogen ions found around your body. We have nitrogen. They're used for proteins and nucleic acids. Some lesser chemicals, calcium. Calcium is used a lot to make up your bones and your teeth. Phosphorus is used for nucleic acids, your DNA and RNA, and ATP, and is required for normal bone and tooth structure. Potassium. There's a lot of potassium that's needed in the body because it helps generate, when we get to what an action potential is, potassium helps generate those action potentials that sends, when nerves what we were talking about in chapter 1 when I talked about when someone touches you, being able to know that someone is touching you. That generates an action potential that sends that information to your brain. We have sulfur. There's vitamins and many proteins in that. Sodium. Sodium is another chemical used to generate action potentials. We have chlorine that helps maintain water balance. Magnesium is needed for enzymes. Magnesium is also a natural muscle relaxer, so it helps kind of relax your muscles. And iron. Iron is found a lot in your blood. It's part of the hemoglobin that helps hold on to oxygen or carbon dioxide. So let's look. What does an atom look like? Or what is an atom even? An atom is the smallest unit of matter. We have protons, which are positive, neutrons, which have no charge, and electrons, which are a negative charge. Both protons and neutrons are found in the nucleus of an atom, while electrons are found in the outside. So let's take a look at some pictures here. The structures of an atom. Here's our nucleus. These are protons in red, neutrons are in yellow. So if we look, there's two different models here. There's an orbital model, which has more of a cloud appearance around the nucleus. Or we have a planetary model that shows the actual electrons. So if we have 2 protons, 2 neutrons, we're also going to have 2 electrons. And our electrons in our first orbit around the nucleus has 2 electrons. The next one contains 8. We're going to talk about some of that. So if you've ever seen a periodic table, every one of the elements on that periodic table has an atomic number and a mass number. The atomic number tells us how many protons are in that atom, while the mass number is the sum of protons and neutrons. Because sometimes there might be more protons than neutrons. There may be less protons than neutrons. So sodium usually has about 11 protons and 12 neutrons. That makes that mass number 23 because we add up the number of protons and neutrons. Isotopes are atoms of an element that have different numbers of neutrons, so they have different mass numbers. Like oxygen, most oxygen atoms have 8 neutrons. There's some that have 9 or 10, but they all have 8 protons and 8 electrons. Most of them are stable, so their structure does not change over time. So there are some actual stable isotopes that have different numbers of protons and neutrons. So a stable oxygen isotope would be oxygen 16, oxygen 17, and oxygen 18. The more stable they are, the less likely they are to change. We also have what are called radioactive isotopes. They're very unstable. They're looking for ways to add more electrons to their outermost ring. So some examples is we have a hydrogen 3, a carbon 14, oxygen 15, and an oxygen 19. Radioactive isotopes tend to decay, and they will actually give off radiation. There's subatomic particles, and then there's also packets of energy. radioactive isotopes they have a half-life, and that's the time required for half of that isotope to decay into a more stable form. So it could be a fraction of a second or millions of years. So radioactive carbon-14 decays actually to a nitrogen-14, but that takes 5,730 years, while 131 iodine has a half-life of 8 days. So they're very different. Some are more radioactive than others. Some have a very long half-life, like carbon-14, and some have a much shorter half-life, like iodine, 131. When we're talking about the mass of atoms, they're technically measured in Daltons. So we know the atomic mass unit of protons, neutrons, and electrons, and those tend to be added up to find what is actually the atomic mass. We know neutrons are about 1.008 Daltons, protons are 1.007 Daltons, and electrons are 0.0005 Daltons. So if we add those together, it's the mass of all the naturally occurring isotopes is the atomic mass, and it's usually pretty close to that atomic number. They don't venture too far away from what their atomic number is for the atomic mass. So as I was kind of saying when I was pointing out isotopes, atoms have a way of gaining, losing, or sharing electrons. That's what helps them become more stable. So an ion is technically an atom that gives up or gains electrons, and this results in either a positive or a negative charge. Ionization is the process of giving up or gaining the electrons. So that's the actual process, is ionization, while the ion is just the atom that gives up or gains electrons. So calcium 2+ is a good example because that is found all over the body. We have calcium 2+ all in a lot of places. They lost 2 electrons. So this 2 means there's 2 more protons than electrons. And that plus means since there's more protons are positive, electrons are negative. This calcium has a positive 2 charge because there's 2 more protons than there are neutrons. When you have an equal number of protons and neutrons, there is no charge because they cancel each other out. But if you get more protons, that atom becomes more positive. If you get more electrons, that atom becomes more negative. So a molecule is when 2 or more atoms share electrons. And it results in a combination of atoms. So if we look at O2, oxygen. Those are 2 oxygen atoms sharing electrons. So the molecular formula is the elements and the number of atoms of each element. So this is telling us we have oxygen elements and we have 2 oxygens. That's what this number is telling us. We also have compounds, which is different elements together. Like H2O is a great example. That's water. We have 2 hydrogen atoms, and we have 1 oxygen. So we have hydrogen, oxygen, hydrogen. That's what makes up water, or sodium chloride, NaCl. That also has 2 different elements to make up a compound. A free radical is an atom or group of atoms with an unpaired electron in the outermost shell. So something like superoxide, we have the addition of an electron to an oxygen molecule. So that's a negative. So if we have an oxygen, we add an electron. We have more electrons than protons, so we have a negative. Now, so what we call this, technically it's a free radical, and its formal name is superoxide. Now, a chemical bond, that is what is used to combine different elements. So a chemical bond is the force that holds together the atoms of a molecule or a compound. So it's the likelihood of forming a chemical bond depends on the number of electrons in its outermost shell. The valence shell is the outermost shell of electrons. So if that valence shell is full, it would have 8 electrons. That is when it's the most stable. If there's less than 8 electrons, they try to steal electrons from other elements. Especially when there's 6 or 7 in that outermost shell, they really want to fill that up. So they want to get those last 1 or 2 electrons to make it stable. So neon is a good example because neon has 8 valence electrons. It has 8 electrons in its outermost shell. So neon will not bond with other atoms. Hydrogen only has 1 electron in its outermost shell. So if we look here, here's my nucleus. I have 1 electron out here. Hydrogen wants to fill this spot with another electron. So that's why hydrogen bonds so easily with many different elements. It wants to fill its valence shell. And for hydrogen and helium specifically, because they're the top 2, they're 1 and 2 on the periodic table, they only have 2 electrons in their valence shell because they only have 1 ring. Helium has both electrons here. So helium's not going to bind with anything else either. We have the Octet Rule. That Octet Rule means it has 8 electrons in its outermost shell. So that's the goal of a lot of atoms or elements, is to fill that outer shell with their 8 electrons. So here's several examples showing how different elements bond more readily or easily with other elements, like hydrogen. Its atomic mass is 1 or 2. It's got 1 proton, 1 electron. It wants to fill this space to have a full valence shell. Carbon Carbon only has 4 electrons in this outer shell. You see 1, 2, 3, 4. Carbon wants to find 4 more electrons to fill its outermost shell. So carbon will bond pretty easily. Nitrogen's another one because it has 5 electrons in its outermost shell. Nitrogen wants to find 3 more electrons. Once it finds its 3 more electrons, it'll be happy. Oxygen has an atomic number of 8 because it has 8 protons, 8 neutrons, and 6 valence electrons. If you see that it has 6 valence electrons, oxygen is used to make water. So for H2O, if we were to put a hydrogen here with 1 electron and a hydrogen here with 1 electron, you can see, this will bond here, this will bond here. Now oxygen has 8 electrons. Hydrogen also shares an electron with oxygen, so now it has 2 in its outermost shell, which makes oxygen happy. Sodium is another one. Sodium tends to give up electrons more than gaining because they have 1 electron in that outermost shell. If there's only 1 electron, they're more likely to give it up to a different element. The more electrons in that outermost shell, the less they're more likely to share electrons. But if there's 1 or 2, they're more likely to just give them up completely. Somebody else take them. Chlorine. They have 7 electrons in their outermost shell. Chlorine really wants to take an electron from somebody. They want that last 8th electron to make them happy, to get their outermost shell completely filled. So chlorine, more times than not, tends to have a negative charge because they steal electrons from somebody. Because then they'll have 8 electrons in their outermost shell. They have more electrons than protons, so it makes them negative. Potassium is another one that is very common to give up this 1 electron in their outermost shell. And as we said before, iodine has 8 electrons in that outermost shell. So they don't really bond, iodine doesn't really bond with any other element because of this. So what do we call those positive or negatively charged ions that give up electrons completely? Because they may only have 1 electron in that outermost shell, or they gain electrons because they only need 1 or 2 more electrons. They're called a cation for a positively charged ion, which means they gave up electrons. They gave up at least 1, maybe 2 electrons out of their outermost shell. So they have more protons than electrons, so they're positive. An anion, on the other hand, is a negatively charged ion. They gained an electron for their outermost shell. Like I said on the previous slide, chlorine is a great example. They have 7 electrons in that outermost shell, so that means they want to gain 1 more. So they gain 1 more electron, so they have 1 more electron than protons, which makes them negatively charged. So these are technically called electrolytes because they are ionic compounds that break apart into positive or negative charged ions in a solution. And remember, your body has a lot of fluid in there, so there's a lot of electrolytes. So hydrogen, sodium, potassium, magnesium, iron, and another iron 2 plus and an iron 3 plus. Those are two different types of iron found in your body, but they are different types of electrolytes that are found in your system. The type of bond that occurs when they share electrons are typically covalent bonds. Covalent bonds are when two or more atoms share electrons rather than gaining or losing electrons. Like when I showed water, we have that hydrogen, oxygen, hydrogen. Oxygen has 6 electrons. Hydrogen has 1. So they tend to share the electron from the hydrogen. They also share an electron from the oxygen on both sides. So they are sharing electrons to make both sets of elements happy, or those atoms happy, by having their outermost shell completely filled. They use covalent bonds to bond the elements or atoms together. So a single covalent bond is when two atoms share one pair of electrons. Like hydrogen molecules, when they share a single valence electron, usually has a single bond. And it's usually shown with just one line. Two hydrogens together share one electron. This is how it's usually written. A double bond is when atoms share 2 pairs of electrons. Oxygen. Remember O2? 2 oxygens together? They will share 2 sets of electrons, or 2 pairs of electrons, so we use 2. Nitrogen is when 3 pairs of electrons are shared. So nitrogen and nitrogen. There's 3 pairs of electrons shared, so we use 3 lines to show that there are 3 pairs of electrons shared. Non-polar covalent bonds, so where one atom does not attract these shared electrons more strongly than the other. So a bond between 2 identical atoms are always non-polar covalent bonds. So when we showed hydrogen, this is a non-polar covalent because one atom does not attract the shared electrons more strongly. Oxygen, same thing. Nitrogen. Now water. H2O, those are not non-polar covalent bonds because oxygen really wants those 2 extra electrons because they cause a more strong bond between the electrons. So those are technically polar covalent. So here's our examples. We have hydrogen atoms, as I showed in the slide above. There's only 1 bar showing the sharing of electrons. Oxygen, they share 2 pairs. Nitrogen shares 3 pairs. We have CH4. They all share. Carbon shares an electron with a hydrogen, with 4 separate hydrogens, and it makes everybody happy. Here's our water. Technically they're sharing 2 pairs. And the element for water is technically written like this, with the bent lines showing how they're sharing their electrons. Our polar covalent shares a lot stronger. There's an unequal pull towards the nucleus of one atom than the other. So it has a partial negative charge near the atom that attracts electrons more strongly. So that electronegativity is the power to attract electrons to itself. Like the bond between hydrogen and oxygen in water. The nucleus of oxygen, like I said before, attracts electrons a lot stronger than the nucleus of hydrogen. So technically, they have a very strong electronegativity and they're a polar covalent bond. Because oxygen wants those electrons more than hydrogen does. We also have hydrogen bonds. Those are different than covalent bonds. A hydrogen bond has a hydrogen atom with a partial charge that attracts a partial negative charge of a neighboring electronegative atom. So again, between oxygen and nitrogen, we'll see hydrogen bonds. Of course, with water, because we have hydrogen bonding with oxygen. So those are technically hydrogen bonds, but they're also considered weak compared to ionic and covalent bonds. Our surface tension is also the measure of the difficulty of stretching or breaking the surface of a liquid. So water has a very high surface tension because water molecules are much more attracted to one another than they are attracted to the molecules in air. So if we're trying to break apart these atoms, surface tension is looked into as well to make sure that, or to see how likely it is to break or stretch those bonds. So here's our example of hydrogen bonds. These are different water molecules, and they have hydrogen bonds between them. So this hydrogen bond in the middle, or water molecule, in the middle has a hydrogen bond to this molecule of water, this molecule of water, this molecule of water, and this molecule of water. So different water molecules will actually form hydrogen bonds between the next molecule of water to help form water that we drink and that's found in our body. So a chemical reaction, if we're trying to form new bonds or break bonds between other atoms, we have what are called chemical reactions. And when we have chemical reactions, we have what are called reactants and products. So the reactants are the starting substance such as hydrogen, 2 hydrogen, and 1 oxygen too. So 2 oxygens already bonded together. The products are what we get at the end. So if we start with 2 hydrogens bonded together and 2 oxygens bonded together. So we have hydrogen and oxygen, and our finished product is actually going to be water. And our metabolism, if we think about our metabolism in our body, our metabolism is the makeup of all the chemical reactions that are happening in our body. So the breakdown of food is chemical reactions, the digestion of, or the absorption of nutrients, those are still chemical reactions all occurring. Those are all a part of what make up our metabolism. So chemical reactions are the form of energy in chemical So if we think, now we're going to look at some physics. The energy is the capacity to do work. So if you're sitting on the couch and you say you have no energy, do you really not have energy, or are you just choosing not to? Because technically, we always have the capacity to do work when we are awake and alert. We have two different kinds of energy. Potential energy, which is our stored energy. So it's energy that's ready to be used as we do work. And then we have kinetic energy, which is our energy in motion. So when you're already moving, like if you're going for a walk or a run, kinetic energy is the energy that keeps you going. We have chemical energy, which is a form of potential energy that is stored in the bones or the bonds of compounds and molecules. We also have in chemical energy, the total amount of energy at the beginning and the end are technically the same. Because if we think about the law of conservation of energy, energy can neither be created nor destroyed. It can just be converted to different forms. So if you ever took physics, you should have talked about the law of conservation of energy. It's just converted, energy's just converted like ATP is a form of energy. And as we use ATP, it gets converted to ADP. Because ATP is adenosine triphosphate. So there's 3 phosphates. And when we use ATP, we remove a phosphate. So then it's adenosine diphosphate. So good example of chemical energy is the food we eat. It's converted into different forms of kinetic energy. So energy that keeps us alert, awake, and moving and functioning throughout the day. And it helps us, as we say, walk, talk, study, do anything you do throughout the day. There's also different types of chemical reactions that require some energy. We have Endergonic reactions. Those require energy, and they absorb more energy than released, while an exergonic reaction releases energy. So it releases more energy than they absorb. So our metabolism in our body kind of couples the two together. So we have exergonic and endergonic reactions within our body's metabolism to help give us the energy to make it through our day. We use glucose molecules. Glucose is sugar, carbohydrates in the body to create energy. And through the creation of energy, we use exergonic reactions to help break down glucose. Glucose can be broken down to create or produce, depending on there's a couple different pathways, but it can create 2 ATP all the way up to 32 ATP. So adenosine triphosphate is ATP. So depending on which pathway it uses, we can create 2 or we can create 32 ATP from one glucose molecule. Our activation energy. It's the energy needed to start some kind of chemical reaction. So it's the collision of energy needed to break the chemical bonds of a reaction. A reactants is the activation energy. So that initial energy is needed to start a reaction, as I said. How much energy is going to break apart the bonds in water? Something to think about. So both concentration of particles and the temperature affect the change of a collision occurring and can cause a chemical reaction. So if we think the concentration, the more particles of matter present in a confined space, the greater the chance of collision. So if we look at, I have this beaker, the more molecules I have in here, the more likely they are to run into each other and cause a collision. If I have a beaker here, there's only a few molecules in there, that's less likely to cause a collision to break bonds. So the more particles, the more likely we'll get some activation energy and break these chemical bonds. Also, the higher the temperature. If the temperature is really high, the particles move much faster. The lower the temperature, the slower they move. So I sometimes, when we heat something up, it's causing those particles to move much quicker, and they're more likely to cause more collisions along the way. So here's a chart that kind of talks about that. We have our potential energy in the progress of a reaction on the bottom, while the potential energy is along the side. If we start, we have the energy of the reactants here. The activation energy is the amount of energy needed to start that reaction. Then after that reaction is started, we see a big decline down. That's the energy released as new bonds are formed and we have our energy of products left. So our potential energy is the energy we possibly have. Activation energies to get started. And then at the end, we have the energy of the products left. So a catalyst. A catalyst is a way to speed up a reaction. It causes things to happen quicker, a lot quicker, because it lowers that activation energy. So we don't need as much energy to start that chemical reaction. And in the body, we have hundreds of different kinds of enzymes. Enzymes are catalysts in the body that help speed up chemical reactions. So in a single catalyst or enzyme can be used for several chemical reactions. They're not going to be used for one reaction and be done. They can be used over and over and over again to help create as many chemical reactions as possible. So here's our example. The red is the activation energy needed without a catalyst or an enzyme in the body. And the blue shows how much lower the activation energy is to get the chemical reaction to start. So having a catalyst makes the reactions much easier to happen. So there's many different kinds of chemical reactions. We have synthesis reactions or catabolism reactions. So synthesis is forming new molecules. So as we form new molecules, and we have atoms, ions, molecules that form even bigger ones. Synthesis, think of synthesis as building, while catabolism is breaking down. So it's catabolism is the decomposition reactions that occur. Like glucose. We break down glucose to ATP, which eventually gets broken down further into pyruvic acid. That'll be covered in Anatomy 2 next semester. So we also have exchange reactions where we have both synthesis and catabolism happening to create completely new molecules. So we have, say we have AB plus CD. They're switching places here. So now we have AD plus BC after we have our chemical reaction. Or we have HCl plus NaHCO3. The two hydrogens bond together with the CO3, and then the NaCl bind together. So these all create new molecules. So we're breaking apart the originals in exchange reactions and making new molecules that the body will utilize for whatever reaction it needs next. Some chemical reactions are reversible, so they can revert back to their original form. Like AB can be split up to A plus B, or it can go back to AB. If we have oxidation, we have a loss of electrons in the process. So in oxidation, we're losing electrons. While reduction is the gaining of electrons. So reduction is gaining electrons. Oxidation is the loss of electrons. So that process to lose them. Then we have oxidation reduction reactions, which is a combination. One of the substances is oxidized and another is reduced at the same time. So a good example in the body is glucose. When oxidized, the energy produced is used to carry out various functions. So as we said, glucose can be broken down into ATP, and we either get 2 up to 32 ATPs, depending on the different pathways, which we'll learn about later. But then the ATPs are used throughout the body to let you walk, talk, think. ATP is used in your brain to process different activities, lets you think, lets you study. You're using ATP just to study. We also have inorganic compounds and organic compounds. So an inorganic compound does not have carbon, and it's a pretty simple structure. It usually includes water, and it could have salts, acids, or bases. So water makes up 55 to 60% of a lean adult's total body mass. It could be more, could be a little less. So if we think about that, 50 to 60% of your weight is water. Only 1 to 2% of that is inorganic compounds. They do not contain carbon in an inorganic, while an organic compound will always have a carbon somewhere. It usually has hydrogens, and it always has covalent bonds. So to be an organic compound, we have to have carbon and we have to have covalent bonds. While that inorganic compound, there is no carbon, and it's pretty simple structure. Organic compounds make up 38 to 43% of the human body. So something to think about. With inorganic compounds, we want a simple structure. Water. That's why water is listed here. Water is an inorganic compound. There's no carbon. And it's a very simple structure. So as I just said, water, it's the most important and abundant inorganic compound in all living systems. Majority, if not all, chemical reactions occur in some kind of watery substance in the body. So the substance is a solvent that dissolves another substance that's called a solute. So there's two different kinds of compounds within water. We have hydrophilic. It dissolves easily in water. If you've ever mixed sugar with water or salt with water, it dissolves very easily. But a hydrophobic, it's not very soluble in water. Oils, fats, if you've ever had water, and here's my little beaker of water. If you were to put oils or fats, they will not mix with the water. You'll see with oil like little droplets in the water of fat or of oil, but they don't actually ever combine. You can try to mix it up. If you mix up that water and then you stop mixing it, oil separates right back out and you see lots of little oil droplets within the water. So as we said, water serves as a medium for most chemical reactions. Chemical reactions, since your body is made up of 55 to 60% of water, majority of those chemical reactions are going to occur there. So different kinds of chemical reactions. Hydrolysis. Hydrolysis is used to break down large molecules into smaller molecules. Like when you eat, hydrolysis, water breaks down the food you eat into some nutrients that are absorbed within the intestines. We also have dehydration synthesis reaction, where two smaller molecules join to form a larger molecule. A water molecule is one of the products that are formed. So if we see dehydration synthesis reaction, we are talking about water being one of the end products. So let's look at water for a minute. Since water is used so much in the body for so many different things, water can be absorbed and released with large amounts of heat with small amounts of change in its own temperature. So water has a very high heat capacity because there's a lot of hydrogen atoms. Water requires high amounts of heat to change from a liquid to a gas. So if you've ever boiled a pan of water on the stove, if you get the water boiling hot enough, you'll see water vapor coming up off of the water that you are boiling. If you put a lid, like a clear lid over that pan, you'll see the water start to collect at the top on that lid. As it turned into a gas, but then it starts to collect again at the top because it has no place to go. So as water evaporates from the surface of the skin, it removes a large quantity of heat. So if you think about it, if you're outside in the middle of a desert, it's a lot of dry heat your body is sweating, but it's also the water's being evaporated so quickly that you don't really know that you're sweating. Versus you're in the middle of a jungle somewhere, very humid, you're sweating and it's just dripping down because the air is very humid, so it takes a little bit more for that sweat to actually evaporate up. So you notice that you're sweating when it's more humid out versus when it's more dry heat. Water is also used in your joints. It helps, it's a major lubricant in the body. It creates mucus. What helps your joints move freely without making any kind of noises is water in those cavities. There's water in your chest, your abdomen, your digestive canal. Water is everywhere in the body. So what is a mixture? A mixture is a combination of elements or compounds that are physically blended together but not bound by chemical bonds. So the air you breathe in every day is a mixture of compounds. Nitrogen is not going to bond with oxygen or argon or carbon dioxide. The air you're breathing in is actually a combination of all these different elements together. So it's a nice mixture. Even though you're breathing out carbon dioxide still goes back in every time you breathe. But you're also breathing in new oxygen that the body needs to deliver to different parts of the body to help move, help make it through your day. We also have colloid, which are solute particles that are large enough to scatter light. So if you've ever driven your car in really dense fog, like really thick fog that you don't see very far in front of you, your headlights are actually, the lights are kind of being scattered because fog is water. Thick water in the air, so it's kind of scattering that light around. So that's what colloid is scattering of that light. Think about the next time you drive through some pretty thick fog with your headlights on. You want to know why your headlights don't shine too far down? It's because the fog is scattering the light of those headlights. Suspension. The suspended material may mix with a liquid or suspending medium for some time, but eventually settles out. Blood is a very good example of that because blood is a mixture of red blood cells and plasma. Blood appears red because of all the red blood cells in there. But after blood is drawn, the tube sits for a while, the red blood cells start to settle to the bottom, while the plasma is more at the top. We talked about this in the last lecture. So the plasma is more of like a yellowish hue to it, while the red blood cells are red, of course. So if the blood sits long enough, it starts to settle out. It's where a lot of times in labs that check blood for people, they use a machine that actually separates the blood out by spinning. And that helps the red blood cells settle to the bottom a lot quicker than if you were just to set the tube in a stand and let it sit for a while. So the concentration, it may be expressed as the mass per volume, percentage, or molecule or moles per liter. So when we're talking about the concentration is what atoms, or if we're talking about blood, how many those red blood cells are actually within that plasma. When you get what are called a complete blood count, a CBC, they're looking at the concentration of red blood cells, the concentration of white blood cells, which we'll talk about when we get to tissues. But all of this stuff is expressed through the concentration of different elements or atoms. So when we have dissociate, it's when inorganic acids, bases, or salts dissolve, they separate into ions and become surrounded by water molecules. So what this is saying is when you put, say, salt in water, salt dissolves in water. So those salt molecules are actually become surrounded by water when you mix that salt with water. An acid is a substance that dissociates into one or more hydrogen atoms in one or more anions. They turn into a proton donor. While a base receives hydrogen from a solution, they're a proton acceptor. pH. If you've taken chemistry, you should know pH is the measure of the acidity or alkalinity of a solution, or the potential of hydrogen. As we just stated above, an acid dissociates into hydrogen atoms in one or more ions, while a base removes hydrogen atoms. So pH is the potential of hydrogen to be removed or to dissociate. So our pH scale tends to go from 0 to 14, and it's based on the concentration of hydrogen atoms in moles per liter. A pH of 7 means a solution contains 1/10,000,000th of a mole of hydrogen atoms per liter. So the midpoint on a scale is 7. That's pure water. Pure H2O. That's right in the middle. It's not considered acidic or basic. It's technically right in the middle. We also have buffer systems throughout our body that help maintain the pH. So if we get some really strong acids or strong bases in our body works to turn them into weak acids or bases. Our blood needs to maintain a pH of 7.35 to 7.45. This is what's needed to keep blood healthy. If it falls below that 7.35, they will actually say that you have acidosis. Or if it raises above 7.45, you'll be told you have alkalosis. So buffers are chemical compounds that convert strong acids or bases into weak acids or bases. So our body has several buffer systems to help maintain the pH where it needs to be. Like our blood, 7.35 to 7.45. Our stomach is more acidic because that's what helps break down food. So different parts of the body have different pHs just depending on where it is and what it's needed for. A carbonic acid bicarbonate buffer system is a common buffer system found in the body. Carbonic acid can act as a weak acid, and bicarbonate can act as a weak base. They can compensate for the excess or shortage of hydrogen. So if we have H plus HCO3 minus, we can make carbonic acid. If you have hydrogen plus bicarbonate, HCO3 minus is bicarbonate. Those combine together to give us carbonic acid, which is where our carbonic acid bicarbonate buffer system comes into play. It makes sure that we do not keep strong acids or strong bases in our system for very long. So organic compounds, we've been talking a lot about inorganic. Let's talk about some organic compounds. They're usually large and carry out complex functions. We have carbohydrates, lipids are fats, nucleic acids, which are proteins, and adenosine triphosphate, ATP. Organic compounds, remember, are held together by covalent bonds. Carbon, as an example, has 4 electrons in its outermost shell, so it usually has a chain of carbon atoms, but can also bind to nitrogen, oxygen, and hydrogen, sometimes sulfur or phosphorus. That chain of carbon atoms that connects together is actually known as a carbon skeleton. A hydrocarbon is many carbons bonded together by hydrogen bonds. We also have functional groups, is where other atoms of molecules bound to the hydrocarbon skeleton. We have macromolecules. Remember, macro means big. So macro are small organic compounds that combine into very large molecules. Macro means large, very large. So when we take organic compounds and create very large molecules, they're macromolecules. Polymers are also large molecules that are formed by the covalent bonding of many identical or similar small building blocks that are also called monomers. The reaction that joins 2 monomers is called a dehydration synthesis. A hydrogen atom is removed from one monomer and a hydroxyl group is removed from the other to form a molecule of water. Carbohydrates, lipids, which are fats, proteins, and nucleic acids are assembled via dehydration synthesis. So we're taking 2 monomers and joining them together in dehydration synthesis. So here's some really good examples of some isomers. Isomers are molecules with the same molecular formula but different structures. This gives us the many different types of sugars. They all have the same molecular formula, but the structures are all different. So a carbohydrate, which includes those sugars, glycogen, starches, cellulose. Cellulose is carbohydrates from plants. They're only about 2 to 3% of the body's total mass, and they function as the main source of energy for generating ATP to drive metabolic reactions. So I said before, sugars. A lot of times sugars are what's used to create that ATP. It's all carbohydrates, really. And carbohydrate actually means watered carbon because we have that's for carbon, and then hydrate, hydro, water. So they function as the main source of ATP that drives metabolic reactions. There's only a few carbohydrates that are used for building structural units, like deoxyribose makes DNA, helps build your DNA. Carbon, hydrogen, and oxygen elements are found in carbohydrates. There's generally one water molecule for each carbon atom. We get monosaccharides, disaccharides, and polysaccharides for carbohydrates. So what's a monosaccharide? It's a very simple sugar. But also disaccharides are simple sugars too. Monosaccharides have 3 to 7 carbon atoms. They tend to end in -ose with a prefix indicating the number of carbon atoms. 3 carbons, trioses. Tri means 3, and then it ends in oses. 4 carbons, tetro. Tetro means 4, oses, tetroses. It's a simple sugar. Pent means 5, oses. Hex means 6, oses, and hept means 7, heptoses. They're telling us how many carbons, and it always ends in that ose. So here's some really good examples. Our pentoses have 5 carbons. Deoxyribose and ribose. Hexoses, remember from above, hex means 6. These have 6 carbons. Glucose, fructose, galactose. They all have the exact same chemical structure or chemical formula, different structures. Same with deoxyribose and ribose. Same chemical formula, different structure. Disaccharides is when a molecule is formed by the combination of 2 monosaccharides by dehydration synthesis. So glucose and fructose combine to form a disaccharide sucrose, or table sugar. And it can be split into smaller, simpler molecules by hydrolysis back into fructose and glucose just by adding water. It's like when you dissolve sugar in water, we are breaking those bonds that brought together glucose and fructose to make that disaccharide sucrose, table sugar. We're breaking it back down to glucose and fructose with just adding water. A polysaccharide. Poly means many. So a polysaccharide contains tens or hundreds of monosaccharides that are joined together by dehydration synthesis. It's usually insoluble in water and they don't taste sweet. So glycogen is the main polysaccharide in the body. It's made entirely of glucose monomers that are chained together. There's a minimum amount stored in the liver and skeletal muscle for energy. So glycogen is more of our stored form of energy. Starches are formed from glucose by plants. It's a major carbohydrate in the diet, like pastas. Do pastas taste sweet? No. Potatoes? Not really, if you eat them plain. They can be broken down into monosaccharides through hydrolysis. So when blood glucose levels fall, cells will break down glycogen into glucose to be released into the blood. So again, that glycogen is our stored energy, and if our blood glucose falls too low, we go to those energy stores to help build up or to help get some energy. So marathon runners tend to eat like a big pasta dinner the night before they race. They're trying to store up as much glycogen as they can to keep running for those 26.2 miles. Cellulose. Those are polysaccharides formed from glucose by plants. Those cannot be digested. Cellulose, your body will never be able to digest cellulose. But that helps build the bulk to help eliminate feces. Lipids, your fats in the body. Lipids are organic compounds that make up 18 to 25% of your body's mass. They have fewer covalent bonds, and they're making most insoluble in a polar solvent such as water. They're hydrophobic. Lipids, remember your fats, your oils. Lipids are the fats. Only the smallest lipids can dissolve in blood plasma. We have lipoproteins, which are soluble because the protein's on the outside and the lipids are on the inside. Fatty acids, triglycerides, fats and oils, phospholipids, lipids that contain phosphorus. Again, if we look at the names, phospho, that's for phosphorus, and then lipids. Steroids are lipids that contain rings of carbon atoms. Eicosanoids are 20 carbon lipids. And then we also have fat-soluble vitamins. Vitamin A, D, E, and K are all fat-soluble. And we have lipoproteins. Fatty acids are the simplest lipids. They're used to synthesize triglycerides and phospholipids. They can be catabolized to generate ATP. They contain a carboxyl group and a hydrocarbon group. Saturated fatty acids have a single covalent bond between the carbon atom and the hydrocarbon tail. So they lack double bonds. There's no double bonds in saturated fats. There's only single bonds. Each carbon atom in the hydrocarbon tail is saturated with the hydrogen atoms. Then we have unsaturated fatty acids that contain one or more double covalent bonds between carbon atoms of the hydrocarbon tail. So if we think about saturated fats, have single covalent bonds. Unsaturated fats have one or more double covalent bonds. So unsaturated fats are not completely saturated with hydrogen. It usually has a kink or a bend at the site of the double bond. We have mono unsaturated fatty acids, has one, mono means one, so one double bond. Poly unsaturated, again, poly means many, so more than one double bond. And the hydrocarbon chain has more than one kink. Here's our examples. Here's a saturated. Palmitic acid. Well, we have a mono unsaturated, has that one double bond in here. Mono means one. Oleic acid. Here's some unsaturated fatty acids. We have palmitic acid again. We can create a water molecule in this the saturated and unsaturated fatty acids. If we have a dehydration synthesis that involves glycerol and fatty acids, we get a triglyceride molecule. Again, we have our one kink because we have our one double bond. Each double bond creates a kink. Triglycerides are the most plentiful in the body. And a lot of times they're known as triglycerols. They can be solid or liquid at room temperature. They're usually saturated fats Oil is a saturated fat at room temperature. And then fatty acids of oil are mostly unsaturated fats. Monounsaturated fats, mono means one again. It contains triglycerides that mostly consist of monounsaturated fatty acids, while polyunsaturated fatty acids contain triglycerides that mostly consist of polyunsaturated fatty acids. Most highly concentrated form of chemical energy is triglycerides. So we get more than twice as much energy per gram as we do carbs, carbohydrates, and proteins. We store our triglycerides in fat, adipose tissue, which is fat, and we have excess dietary carbs, fats, and oils that are also stored in adipose tissue. Phospholipids. They contain two nonpolar fatty acids, and they can only interact with other lipids. So we have amphipathic, which are molecules with both polar and nonpolar parts. They line up tail to tail with a double row to make up much of the membrane that surrounds cells. So here's our example. We have a polar head and the nonpolar tails. So here's an example of a cell membrane. We have our heads on the outside, polar heads, while the nonpolar tails. The polar heads are water soluble while the nonpolar tails are not. This helps protect the cells and keeps them safe. We also have steroids. Steroids are 4 rings of carbon atoms. The body synthesizes steroids from cholesterol because it has a large nonpolar region. Steroids are used for hormones in the body. Sterols, which is a form of steroids, have at least one hydroxyl group. Hydroxyls groups have an OH. Cholesterol is a steroid. Estrogen, other sterols, testosterone, cortisol, bile salts, and vitamin D. Those are all sterols. Remember, your skin makes vitamin D from sunlight. So here's some examples. We have our hydrocarbon tail. This first one here is cholesterol. Well, we have estradiol, which is an estrogen, or female sex hormone. Here is testosterone, the male sex hormone, and cortisol. Estradiol has that hydroxy group, testosterone has that hydroxy group, and cortisol has that hydroxy group. So they are all sterols. Eicosanoids is another lipid that's derived from a 20-carbon fatty acid chain. Arachidonic acid is a good example of an eicosanoid. We also have prostaglandins. Prostaglandins will modify the response to hormones. They contribute to the inflammatory response. So when you get hurt, prostaglandins are what are used to inflame the area. They prevent stomach ulcers. They dilate large airways to the lungs, so they make the airways bigger. Prostaglandins help to regulate the body temperature. They influence the formation of blood clots. We also have leukotrienes. Leukotrienes are big in allergic reactions and the inflammatory response. So when you have an allergic reaction to something, leukotrienes are what help participate to create that allergic reaction. Proteins are large molecules that have carbon, hydrogen, oxygen, and nitrogen. Sometimes sulfur. Your body has about 12 to 18% proteins in your body mass. They help contribute to body tissue structure. There are also enzymes that are proteins. As we talked about with catalysts, enzymes speed up chemical reactions. Some of those proteins are used as enzymes to speed up chemical reactions. There's also hormones that are proteins that are used throughout the body. Amino acids. Amino acids are monomers of proteins. So they're parts of proteins. There's technically 20 different amino acids. They all have a hydrogen atom and 3 functional groups attached to the central carbon atom. Amino acids chain together to form proteins. So as proteins get broken down, we're creating amino acids because those amino acids are what bond together to make those proteins. There's different kinds. There's a peptide bond, which is a covalent bond between amino acids that remove water. We have a dipeptide, which contains 2 amino acids. Tripeptide, tri means 3. And it's an amino acid that attaches to a dipeptide, so that gives it 3 amino acids, or a polypeptide, a chain of amino acids together. So here's some examples. We have a non-ionized form of an amino acid, while we also have a double ionized form of an amino acid. So if we look, we have 2 hydrogens over here, 3 hydrogens over here. This nitrogen has a positive charge, but then we have an extra hydrogen on this side that we don't have over here. That's what makes a non-ionized versus an ionized. Then we have glycerin, cysteine, tyrosine, and lysine. Those are different kinds of amino acids that are found naturally in the body. So if we look at the structure of proteins, their primary structure is technically one-dimensional, flat, and they're genetically determined. So in something like sickle cell disease, if you've ever heard of that, it affects the red blood cells. It's a mutation in amino acids. There's 2 mutations that carry the oxygen protein hemoglobin. And what happens is these red blood cells get a sickle shape. So all the red blood cells start to get this shape. The problem is with sickle cell, these red blood cells can't go through the really small capillaries, which are the smallest type of red blood cell. They get stuck, and it starts causing problems. Proteins also have a secondary structure. They have a 2-dimensional twisting or folding of neighboring amino acids in a polypeptide chain. So they have alpha helixes and beta pleated sheets. Secondary structure, think secondary 2, 2-dimensional. Primary, like first grade, first, 1-dimensional. We also have a tertiary structure, or a 3-dimensional shape. Remember before, tert means 3. So some may be attracted to water while others repel it. We have a hydrophobic amino acids. They're usually at the core. They're away from the surface because they don't like water. They stay as far away from water as they can. We have fibrous proteins. Those are insoluble in water. They have a polypeptide chain that forms long strands that are kind of parallel to each other. Like collagen. We'll get to look at some collagen when we get to tissues in chapter 4. They're also in bones and ligaments. Elastin. That's what helps your skin stretch. Blood vessels. It also helps those stretch. Keratin. Gives your hair color. Nails. Dystrophin. In muscles. Fibrin. That helps form your blood clots. And actin and myosin are also for fibrous proteins. Those are what help muscle contractions. We have globular proteins that are mostly soluble in water. We have peptide chains that are spherical, and they have metabolic functions as well. Globular proteins are like enzymes, antibodies that help keep your immune system. We have complement proteins. Those help protect against diseases. Hemoglobin, lipoproteins, which are transport lipids. Albumin, which helps contribute to the blood pH, and insulin. So here's some examples of the different types. Primary structure, again, is our flat amino acid sequence. We have an alpha helix, kind of circular, or a beta pleated sheet. We have a tertiary structure and a quaternary structure. So there's many different shapes that these proteins can make. Proteins tend to denature. So that's the unraveling due to the alteration of the homeostatic environment. When proteins denature, they're not functional anymore. Some can be reversed, but most of them cannot. Think about when you fry an egg. An egg starts as a liquid. You fry it, it becomes kind of solid. The white becomes solid when you fry an egg. That's a permanent denaturing. Once you fry that egg, you cannot undo it. Enzymes. Other stuff found in the body. Again, we've talked about enzymes. They're catalysts. They tend to be made of proteins, and some are made of two parts. We have an apoenzyme, which is a protein portion, and a cofactor, which is the non-protein portion. It could be a metal ion or an organic molecule, which is a coenzyme. Coenzymes tend to be derived from vitamins, and they usually end in ACE. And a lot of times they're named for the chemical reaction that they catalyze. Oxidase, oxygen, so they add oxygen. Kinase adds phosphate. Dehydrogenase, that D tells us it removes. So it removes oxygen. Anhydrase removes water. Protease, pro for proteins, proteases break down proteins. Lipases breaks down triglycerides. Triglycerides, again, is a form of fat. Lipid, lipase, lipid is a fat. So enzymes are very specific. They can only bond to certain places, certain substrates. So the site of the reaction is the active site. So it's like a lock and key. Like a key can only open a certain lock. The same key cannot open every lock in the world. So enzymes are exactly like that. They can only open up a specific lock. They can only activate a specific chemical reaction. They're very efficient too. They work to speed up reactions from 100 million to 10 billion times than the site without an enzyme. So if an enzyme didn't exist, the reaction would be 100 million or to 10 billion times slower. They can convert between 1 and 10,000 molecules in 1 second. So they are also subject to a variety of cellular controls. So their rate of synthesis and concentration at any given time are under the control of the cell's genes. They have both active and inactive forms, and they're determined by the chemical environment. So as we've said before, enzymes tend to lower that activation energy for that chemical reaction. It makes that chemical reaction a lot easier to occur, and it doesn't require as much energy to occur. So it helps to bring the substrates together for the reaction to occur. Substrates make contact with that active site, and they form an enzyme-substrate complex. So these are the steps to that reaction. That substrate molecules are transformed by rearranging existing atoms. Then after the reaction, the unchanged enzyme is free to attach to another substrate molecule and repeat the same reaction. So here's an example. Our enzyme bonds with our active site here. That enzyme catalyzes the reaction, and then when the reaction is complete, that enzyme leaves to go attach to another substrate and repeat the same reaction over and over and over again. So here's a molecular model of an enzyme and substrate. We have our enzyme, hexokinase, which is this really big structure here. And our substrate is glucose. When glucose and the substrate, or that enzyme, and that glucose molecule come together, it breaks down that glucose, and then that enzyme remains unchanged, and it goes on and binds to the next glucose molecule. Now let's look at nucleic acids. A nucleic acid is your DNA and your RNA. They are repeating monomers that are organic molecules that contain carbon, hydrogen, oxygen, nitrogen, and phosphorus. Deoxyribonucleic acid, that's DNA, that's what DNA stands for, deoxyribonucleic acid, that's what you inherit from your parents. And that information is inside every human cell in the body. A gene is a segment of a DNA molecule. We take that whole DNA molecule, we take a segment of it, just a part of it, that's a gene. Break it up. And those are the different genes that tell different cells in your body how to replicate, how to do things. Ribonucleic acid is RNA. And it relays those instructions from the genes to guide each cell's synthesis of proteins from amino acids. So RNA is what tells the body how to make more proteins, how to make specific, any kind of gene for whatever's needed in the body. So DNA has 3 different parts. It has the nitrogen base, which is either adenosine, thymine, cysteine, or guanine. Those 4 nitrogen bases are either purines or pyrimidines. Adenosine and guanine are the purines. They are large double-ringed bases, while the pyrimidines are thymine and cysteine. Those are smaller rings. We have pentose sugar in our nucleic acid, which pentose, penta, 5 carbon sugar. Deoxyribose is attached to each DNA base. And then we have a phosphate group. Alternate with pentose sugars to form the backbone of the DNA strand. Then DNA has a double helix. It's 2 strands that form a spiral ladder. So if we look, we have our phosphate group, another phosphate group. They bind to the deoxyribose sugar to have thymine, adenosine, those bond together, or cysteine bonds with guanine. And they come together to make this DNA molecule. It looks just like a ladder that spirals up. RNA, on the other hand, it's a single strand of DNA. And instead of thymine, RNA contains uracil. They still bond the same, but instead of thymine, it contains a uracil molecule and base instead. There's 3 different kinds of RNA. There's messenger RNA, ribosomal RNA, and transfer RNA. They're all used to help create different proteins, different parts of cells, and tells your body how to function. Let's quickly look at ATP. ATP, adenosine triphosphate, is known as the energy currency of the living systems, because without ATP, we have no energy. So ATP, it transfers energy in catabolic reactions, breakdown reactions, to power cellular activities that require energy. So writing, taking notes, that all requires your ATP. So the enzyme that catalyzes ATP is ATPase. ATPase, that's an enzyme. And then the reactions that move the 3rd phosphate group creates, as I stated earlier in this lecture, adenosine diphosphate, ADP. By removing that phosphate group, that produces the energy. The process of removing that phosphate produces energy for you to do something. We have that anaerobic phase, and no oxygen is needed. That is where the glucose is broken down to create 2 ATP. While the aerobic is where we need oxygen. That is where we create 30 to 32 ATP and we create heat. So if you're exercising, doing aerobic type activities, you are taking in a lot of oxygen, and that is allowing glucose to be broken down into carbon dioxide and water, along with 30 to 32 ATP. If you are sprinting as fast as you can, that's anaerobic. So that's not using oxygen, and that's only creating 2 ATP per glucose molecule, because you can't sprint very far. Most people can sprint short distances. Versus aerobic, you can go a lot longer because you're using oxygen. And that is all for today's lecture on the different types of chemicals, chemical bonds in the body. Thank you.